Good morning, everyone. Welcome back. We are in the course on fundamental principles in bioethics in the Masters of Bioethics here at St. Thomas University, beginning as always with a little prayer in the name of the Father, the Son, and the Holy Spirit. O Lord, send forth your Spirit, and we shall be recreated. You shall renew the face of the earth. Amen. Father, Son, Holy Spirit. All right, again, apologies for the delays. When it rains, it pours. We had a number of issues, but we're rolling now, and hopefully Jorge and Chris can also hear me and see the screen.
Yes.
All right, thank you.
Yes.
All right, so any questions or comments from previous lectures or the program, anything? We're okay? All right, so let's see. We have been looking at some of the fundamental laws for evolution. First, the evidence for evolution from various sources. The fossil record is the most obvious thing, that these are living organisms, or fossilized organisms that were living at some point, typically millions of years ago, and they look different but similar to some of the living organisms. So it stands to reason that there is some transition there from these ancestors, okay? And also the best proof really is what we call the phylogenetics. What is phylogenetics? Phylogenetics, or the molecular evidence for evolution. Genetics is a reference to the DNA, the makeup, right? Even the fact that the DNA is the universal molecule of inheritance. Did it work? Jordan, your internet? Okay. Rogelio, Carolina? You okay? All right. Okay, Carlos, thanks again. All right, so, right, the molecular evidence. First, the fact that DNA is the universal molecule. In other words, what a coincidence, let's say, that all of the living organisms that we see today, their inheritance is transmitted through this molecule, right, which is a macromolecule. Is DNA a small or a large molecule?
Large.
Exactly. Obviously, it is macroscopic, supramacroscopic, because it's a molecule, but it's a molecule that contains millions of atoms, literally, the twisted ladder that we are familiar with. And so it stores information. It stores a lot of information, right, at the chemical level. And today, of course, through biochemistry and analysis, the genetic code has been deciphered. And we have a very elegant transition from DNA to the messenger RNA to the actual protein sequence, the sequence of amino acids that corresponds to 3 bases from the DNA sequence. And so all that already has been elucidated, called elucidated, meaning that it has been deciphered. The genetic code and all the transition to making the proteins in the cells, again, hundreds of thousands of proteins. That is called the central dogma in molecular biology. Central dogma, from DNA to messenger RNA to protein, right? Transcription, translation are the 2 processes. And in luxury of detail at the molecular level. So it gives a mechanical basis for metabolism, a mechanical basis for inheritance. We used to have here at St. Thomas University a physics professor, excellent person. He was a graduate from MIT, astrophysicist, and he went on to run the astrophysics department of NSF, National Science Foundation, in DC, or now they're in Maryland, in Virginia, I'm sorry. At any rate, we would have very interesting conversations because he was also a devout Catholic and talking about the relationship between, of the interaction between science and faith, science and religion. And he would tell me, Father, it's amazing how mechanical the whole thing is of the universe. And when we get down to the smallest level or the largest level of scale, those universes, right? The microscopic, the super microscopic universe and the super macroscopic level, it's all very mechanical. In other words, there is an explanation. There's a mechanical explanation, just like we can explain the movement of a car through the mechanics of all the parts that work in it. The same with life, the same with life. At the biochemical level, it's really a panoply, a sophisticated level of reactions, of chemical reactions that are occurring. We call them cascades. In other words, one chemical reaction leads to another. So the product of one chemical reaction becomes the starting element of the next reaction and so forth. And the vast majority of these reactions, chemical reactions in the cell, at the cellular level, are done by the proteins. The proteins, many of which are enzymes, precisely. Enzymes catalyze reactions, right? They regulate chemical reactions in the body. Reactions that would normally take a long time, enzymes speed up chemical reactions, essentially, and that's what we call metabolism. So when we put the whole sum of metabolism at every cell in our body that is alive, then that is life. That is the life of the organism itself, whether it's a single cell or trillions of cells like we have and many other organisms, okay? All right, so this phylogenetics, we can see that there is a branching effect known popularly as the tree of life. So the tree of life will have many, many branches at the periphery. But if we go down those branches into the main trunk and eventually into the root system, it points toward a common ancestor, which is interesting and kind of bewildering to consider that all of the life that we see today, these 2 million species that we have already discovered, and the estimated 5 to 10 million species that are still out there, maybe more, they all came from one single species and one single organism and one single cell, which was obviously a unicellular. And that even could have come from inorganic material, as I was talking about the origin of life from an organic level or from a chemical level in last lecture, all right? So it's pretty humbling. Huh? Now, of course, for the human, there is an added, there is a plus. And essentially, what is the plus in the human that would distinguish us, even at the organic level, from all the other species around, the closest ones to us, like I've mentioned today, the chimpanzee, and the furthest away, maybe some bacteria or archaea, I guess, would be the furthest species from us, right? What do we have? What is that plus? Beyond the organic, what do you think, Jordan? Bilateral symmetry or bipedal? You think of bipedal? Yes, fine. It is bipedal at the organic level, but I was thinking more at the intellectual and spiritual level. The conscience, the will that volition that we can determine, we can decide what to do. Of course, you know, natural psychologists and evolutionists tell us that we probably are less willing to admit that most of our behavior is pretty deterministic already. So we may think we are exercising free will. Actually, a lot of our behavior is fairly hardcore into some pattern of behavior, right? For example, fear, fright, and things like that, when it's spontaneous, typically it is some organic process taking over, all right? But I don't think that we can deny that we have also a will to decide when we reflect upon things, to decide one way or another, or even a multiple of decisions on any particular topic or circumstance. So that really is extra. And so that just introduces me into what I'm going to be talking about today, which is mostly chapter 5 of Meyer, which is on variation, because to get to the punchline, evolution by way of natural selection, remember the title of Darwin's thing, by means of natural selection, right? The origin of species. Selection acts on variation. Remember that little phrase, okay? Or sentence, because it's a verb. Selection acts on variation. You'll see what I mean by that further as we go along in the lecture and in the following lectures. But really it is because of variation within the species. Okay, for example, we're all human beings, we're all Homo sapiens biologically, all right? And yet there is variation among us, between us, at these 4 levels, the level of morphology, physiology, molecular, and behavioral variation, even just among the 6 of us who are in the lecture today, all right? Okay, so, but before moving away from chapter 4 of Meyer, which I recommended that you read, but I don't need a summary of it because I think it's, with all due respect, faulty in this sense. It talks about a lot of isms there, and basically that chapter is devoted to saying that philosophical and religious beliefs slow down the progress of science. And I guess historically it's a proper claim. However, the next step that he takes is, because of that, he essentially rejects philosophy and religion to play any role in human knowledge, and he just goes straight into biology and evolutionary biology and so forth, but precluding any possibility of dialogue or contribution from philosophy and or theology to the progress of science today. So just want to leave you with this thought. He talks about a lot of isms, all right, a lot of isms. And the main ism that he rejects is this essentialism, which he associates it with the philosophy of his time and also the religion of his time and for several centuries before that. So that is his claim that this mentality, which was widespread among the population, especially the European population at the time, slowed down the progress of science to the point of even condemning some scientists like Galileo and Copernicus and a number of the scientists who were emerging in the Renaissance in Europe at that time, all right? And all the skepticism that went on with Darwin, and even the ridicule of Darwin looking kind of like a monkey because he was claiming that we descended from the monkey, or at least they thought that he was claiming that, which I want to mention also. I'll talk about contemporary ancestors. We cannot have contemporary ancestors. That's a contradiction in term. Either they are ancestors or we are contemporaries, okay? And because the monkey and us both exist at the same time today, then neither did we derive from the monkey, nor did the monkey derive from us, because we are contemporary. Now, we both have common ancestors, and that's a very different story, right? Just like we have a common ancestor with the fly and with bacteria, way back. Any rate, what I think is his mistake is that he doesn't have really, to my knowledge, a systematic philosophical background, or theological even less, especially in Catholic philosophy and theology. I'm talking mostly about Thomism and classical Thomism developed by Thomas Aquinas, and theologically, and himself, Aquinas, going into classical Greek philosophy, mostly Aristotle, for his philosophical underpinning. of his theology of Aquinas, okay, which is what is known as Thomism. But basically, to distinguish between essence and accidents. And this is even in our language to this day and in many languages of the world, what we call the noun and the adjective, if you will. The noun, another word for the noun is the substantive in grammar, el sustantivo en español. And the noun, what is the noun? For example, who are we? We are men and women, okay? So that is the noun, we are human. So that noun responds to the question, what question? The response to the question of what? What is it? What is it? Quid est, what is this? Oh, phone. Well, it's not really a phone. I know what a real phone looks like. It's a plastic box with a handle on it, with a wire, had a rotary thing on it. This is actually a microcomputer. This is the real mini computer, right, in our pockets. But it tells us what the thing is. This is a computer. This is a screen, etc. This is a room. The noun tells us the essence of things, the whatness. And then the adjective qualifies that noun. Those are the accidents. A glass could be a glass made out of glass, or it could be a glass made out of plastic, or it could be a glass made out of metal or wood, but it's a glass holds water, has a particular function. And so we see that the noun tells us the essence of things. And that's where I believe, again, in respect to Meyer, the great mind that he was, he confuses essentialism with the essence, all right? He rejects the essence of things because of the exaggeration, and typically an ism is an exaggeration. But that's where I think he misses the point. And then the adjective will qualify the noun. Those are the circumstances. Whether the person is tall or fat or old or ignorant, those are adjectives. But it doesn't detract from the fact that that is a person, a human being, which is the core, the substance, the essence, all right? And so it's important for us to understand these concepts because later when we talk, for example, about the beginning of human life, it's going to be difficult to see a single cell as fully human, the zygote, the fertilized egg, right? But we need to get there. And it's only with the proper use of philosophy and reasoning that we can see a single cell as a human being, as human as you and I in essence. But of course, at the very early stage of development. Even a baby is not fully developed either, right? Okay, so that's where I'm going to leave it. You can read it again if you want to, but keep in mind that as far as I can tell, he didn't have a systematic upbringing in philosophy, at least in Thomistic philosophy, which is the one that we use in Catholicism today. And it is based also on what we call natural law, that we allow nature to tell us the function of things. That's another principle, if you will, that we look at when we analyze nature in bioethics, is that form follows function. Form follows function. The structure of the thing is according to the function of the thing and not vice versa. For example, if an arm is meant to bend so that the hand can reach the mouth, for example, then it better have a hinge somewhere along the way. Otherwise, it's just a long stick that I cannot get close to my mouth, all right? So if the hand is meant to reach the mouth, then the arm better bend at some point. And that's what we call the elbow, you see? So the structure of the thing is according to the function of the thing. And also, while I'm on that, I can see, you can see the difficulty that scientists, that empirical scientists have today because they typically lack a philosophical background. And belaboring the point here a little bit because I want your minds to expand and to think critically, all right? For example, PhD. I have a PhD in genetics. What does PhD stand for? It's an acronym. It's a contraction of what? Philosophical doctorate. That's what it means. PH is for philosophy and D for doctorate. It means a philosophical doctorate. I have a doctorate of philosophy in genetics. Basically, it means what? That I can philosophize about genetics. That's what the PhD means. The PhD is that when a person has a PhD, that person can philosophize about that. Sorry, it's not the class here. You have to look somewhere else. Maybe upstairs, okay? You're welcome. The nursing program. Okay, so you see, a PhD means it's a terminal degree. It's the last degree in academia, right? Meaning that that person is supposed to be able to philosophize about the subject matter. Well, I can tell you, because I've been in science for decades now, even before the priesthood, and for decades, I have been trying to have philosophical arguments with my colleagues, my empirical colleagues in science, and it's very difficult because they really don't know, with all due respect and humility, they don't know how to do philosophy. Most of them don't. They're excellent empirical scientists, you know, it's almost, they're excellent technicians. They know the techniques in the lab much better than I do, for example, okay? But they don't know how to philosophize about nature because they haven't had the background. Because today, empirical science, there's so much data and so much knowledge and so many pathways that the students have to learn, all right, even up to the PhD level, that they don't have time for the philosophical courses. And so what happens at the end of the day, these people who are very bright and are running multi-million-dollar grants throughout the US and throughout the world, they end up with the thought that philosophy is an opinion. And so if you have your philosophy, I have mine, and it's your opinion, and I have my opinion, and the best that we can agree is to disagree on opinions. But no, absolutely not. Philosophy does have a method. It's a method of critical thinking, which is basically logic and syllogisms and so forth, and reasoning. And so there is a method, and so it's a science, but it's not an empirical science. It's a metaphysical science, right? And so continue to ponder these things, but when real, true philosophy and theology is applied to nature, then it complements the picture for empirical science and for the data of science, all right? So far from being in opposition to each other, even in last cohort that is now graduating in December, one of the students showed me a film about some fundamentalist group that after half an hour or more of this film going on and denying the fossil record, essentially saying that these are animals that died recently, you know, a few years ago, even finding tissue within these fossils and so forth. At the end of the day, the conclusion is that you either accept God and faith and religion, or you accept evolution, all right? And never shall the two meet. Well, with all due respect, that is a fundamentalist view of religion, which is a literal interpretation of the Bible. And we as Catholics take a big exception to that kind of analysis, all right? Because we find it extremely narrow-minded and full of contradictions, as I will show you in more detail toward the end of this course, in the second half of the course. Okay, so that's enough for my 2 cents there with regards to the value of philosophy and theology when it's done properly. Let's get back now to variation. And variation, keep in mind, this was kind of where we left off last time, is at these 4 levels, okay? At the morphological level, at the physiological level, at the molecular level, and at the behavioral level. Let's reflect that a little bit for a moment. Basically, the first 2 are structure and function. Morphology is the shape. Oh yes, that's another thing. When I say this, a dictum that you hear in science and in biology and in nature, form follows function. But you see, there's another error of empiricists. Form, right, for a natural scientist or for an empirical scientist. You understand what I say, what I mean when I say empirical scientist, right? One that is measuring physical realities, material, anything that can be measured. All right, so for empirical scientists, form essentially is the same as shape. What they're saying, when they say form, they really mean shape. So not what is the form of this form, but what is the shape of this form, okay? What is the shape of this room? What is the shape of a structure like a muscle? But in philosophy, form and shape are very different, you see, because form is the essence of the thing, and shape is the physical reality. So for example, how many shapes do we have in this room? Sorry, guys, for being online, but we are physically present here. So in this room, how many human shapes do we have? All human shapes, right? But how many human forms do we have? We have one human form, which is humanity. We all share a common humanity. So the form is speaking of the essence of things. That's the real form, all right? And so we share one form, which is the essence, which is humanity. So again, with all due respect, empirical scientists don't get it. When they mean form follows function, they actually mean shape follows function, structure follows function, right? And not vice versa. So morphology is actually shapeology. It is a whole science that studies the structure of things. Typically, for example, for PT, for a physical therapist, she or he better know the morphology of the body in great detail, all of the muscles, all the tendons, ligaments, bones, etc., because that's going to be their profession. Okay, physiology is about function. Physiology is the pathways that we're talking about at the cellular level, the tissue level, what the different organs and tissues do, right? That's the physiology, the function. Remember also, another one of my little sayings, normally we see and we experience structures. We don't experience functions. We have to reflect a little bit. What does this do, right? What does this do? That's the function, because it's not, sometimes it's not very apparent. All right, and the extreme, kind of the extreme example of this, that function is not very apparent, is what we call mimeticism, mimetism in nature. For example, a large moth having two circles at the lower wings, and those circles are very similar to the eyes of an owl, to the open eyes of an owl. But obviously, it's not an owl, it's a moth. All right, mimeticism, different function. It's a fake, right? It's to distract a potential predator, thinking it's an owl, and it's not an owl, it's a moth. All right, so at the molecular level, well, again, the pathways, we see variation, so that even among the six of us, okay, including Chris and Jorge, our metabolism is, in the big picture, we have the same metabolism. Basically, we have the metabolism of a human body, but at the level of detail, there are going to be variations at the molecular level of the six of our metabolism. For one thing, we all have a slightly different age, and that already differs the metabolism a little bit, et cetera, et cetera. Okay, so at the level of detail, molecular metabolism, molecular structures and functions are also going to vary within the same species, between different individuals. Behavioral, I think it begs the question at the behavioral level, again, given with any particular situation, any one of us can react in any number of ways. That is not exactly the same way. So there's also variation at the behavioral level between individuals of the same species, okay? And so that's where selection is going to act, on the combination of these four variations, of these four types of variations. Okay, then we're going to look at a little bit, let me get back to the PowerPoint. What is the population and population range? Because within the range is where we're going to see the variation in the range, all right? We're going to drill into how these ranges come about, how these variants come about, which is something called polymorphism, which for looking at polymorphisms, we need to do a little bit of genetics. Then we're going to get into quantitative traits and Darwinian evolution, all in the first half of this lecture, because then in the second half, we're going to look at 3 of the 4 main forces of evolution, which are mutation, migration, drift. And then next lecture is going to be all on natural selection. All right, so that's for the second half. Going forward, if there are no questions or comments, let's get into it. All right, so variation at these 4 levels. Okay. Well, let's look at twins. If I'm able to prove to you that twins have variation, their variance between identical twins, all right, then at these 4 levels, okay, consider what I'm saying. What I'm going to do is I'm going to prove to you, give you the proof that there is variation among or between identical twins of 2 humans, right? At these 4 levels, in these 4 areas. So if there is variation between identical twins, which are the closest humans, they're essentially clones of each other, then there's obviously variation among every other individual of that same species, right? Because that would be the closest identity, morphological, genetically. So let me introduce, first I need to explain then identical twinning, just to review, I don't know if we cover it or not, but there are 2 possibilities of twinning for the human and for mammals and animals in general that reproduce sexually. Let's stick to mammals for now, either fraternal or identical twins, okay? Now, these are popular terms, fraternal and identical. For the sake of science, fraternal twins are called dizygotic, and identical twins are called monozygotic. Again, the words are important, they have meaning, and the meaning here is dizygotic, the prefix di means 2, exactly, and zygotic is a reference to the zygote, which is the first cell of a new individual, whatever species. It happens at fertilization, at the completion of fertilization. So dizygotic meaning that there are 2 zygotes, essentially, whereas monozygotic, there is 1 zygote, okay? So how do we get there? First, look at dizygotic to then discard it. For dizygotic, we have 2 zygotes, meaning that there were 2 eggs, 2 ova that were fertilized by 2 sperms. So what happens? Normally in the human, we know that a woman naturally and normally produces 1 egg, matures 1 egg, 1 ovum per cycle on an average of 28 days, et cetera, okay? So there's 1 mature egg there that normally is in the ampulla of the fallopian tubes for about 24 hours, and that's the narrow window of fertilization is 24 hours. After that time, normally that egg becomes over-ripened and is discarded, and is discarded through the process that we know as menstruation. All right, so the egg is there, but for an ejaculate to be considered functional, to be considered potent, capable of fertilizing, so in principle, how many sperm is needed to fertilize 1 egg? 1 to 1, right? 1 sperm to fertilize 1 egg. But we know that functionally, clinically, for a human ejaculate to be considered functional, capable of fertilizing, you know how many sperms it needs to have? It needs to have between 100 to 150 million sperm. It is amazing. That is a very strong selective pressure against human sperm, all right? And so that's why on average, it is the strongest, most potent sperm that fertilizes that egg, which makes for the most vigorous child possible, just from natural selection. So there's tremendous selective pressure, intense selective pressure against the sperm for motility and capacity and vigor and all that. At the end of the day, on average, in any normal average intercourse, there would be millions of sperm floating around or swimming around, and so whatever egg is there is going to get fertilized, right? So if a woman happened to mature 2 eggs, one from each ovary, let's say, or both from the same ovary, but there are 2 eggs sitting there, on average, they're going to get fertilized. And they're going to make 4 dizygotic twins. Now, because we know also that it is the sperm that establishes the sex of the child, right? The Y chromosome or the X chromosome in the sperm, then for dizygotic twins, we can have 1 boy and 1 girl, or they can both be boys, or they can both be girls. All 3 possibilities. Different for monozygotic twins, very different. In monozygotic twins, there's only 1 egg produced in that zygote. There's only 1 sperm that fertilizes that 1 egg. So there's only 1 zygote, monozygotic, all right? And then within the first 2 weeks of embryonic development of the human, that 1 zygote can split into 2. It can happen all the way from the first mitosis, which is again within 24 hours. That zygote will split into 2 and become the 2-cell stage. Then each one of those 2 will split again into 4, 8, 16. You know, it's a geometric progression. So at any stage between the first 2 weeks, an implantation occurs in the first week, 5 to 7 days after fertilization. But even after implantation, in the gastrula stage, within the first 2 weeks in the human, it's known experimentally that the embryo can split into 2, all right? And that is what causes monozygotic twins. And so monozygotic twins, can they be of different sex? Can one be a boy, another the girl? No, why? Because there's 1 sperm, right? And 1 sperm is either X or Y chromosome. And so monozygotic twins necessarily have to be the same sex. And sometimes they can split again. You can have quadruplets. It's much more rare, but twins, monozygotic twins, is not that rare. I remember one time reading about it when I was doing my first doctorate. It's 1 in 64 pregnancies, 1 in 64 human pregnancies. It's actually monozygotic twins. It's not all that rare, you know, even less than 1 in 100. At any rate, I want you to meet here now Claire and Laura Newing, who are monozygotic twins. Okay? So just from this photograph, which I found at the internet, I'm going to prove to you from observation, critical observation, that these 2 identical twins have slight differences, morphologically, physiologically, molecularly, and behaviorally, okay? So let's go forward. Here they are in this photo. Let's look at morphology. I'm proposing to you that they, the femur of one of the 2 is a little longer than the femur of the other. The femur is the longest bone in the body, and the femur is mostly responsible for our height, okay? Because the femur is the longest bone, so if it stretches out 1 more inch, then we're gonna be 1 more inch taller, and so forth. It's a variation, right? Height. We don't all have the same exact height, right? Okay, so just from the photo, who seems to be slightly taller than the other? Claire or Laura, right? I put the end of the photo there on purpose so you can see that she's maybe 1 or 2 inches taller. Claire is 1 or 2 inches taller than Laura. I don't know exactly, yes, she could be wearing high heels, right? But assuming that they both have the same type of shoes, it looks like Claire's femur is a little longer than Laura's femur, which will be a slight difference in morphology, okay? How about physiology? What causes freckles? Where, so how to phrase this, where the freckles occur on the skin, right? On the epidermis, the outer layer of the skin. And those epidermal cells, they have a pigment inside, right? What's the name? Exactly, melanin. Melanin is the pigment that causes our skin to be of a tan color, all right? So for example, Norwegians have very little melanin on average because they look very pale, all right? whereas Africans in general have a lot of melanin, right? Because they're very dark in their skin. And it coincides with what? It coincides with the angle of incidence of the sun rays. You can translate that into the UV that is penetrating. So the UV goes through less atmosphere at the equator because the rays are more perpendicular to the surface, to the tangent of the Earth. And it goes through less atmosphere, therefore less UV is filtered out at the equator than at the poles. Because at the poles, the rays of sun are not coming perpendicular to the tangent of the circumference, but they're coming at an angle. So they go through more atmosphere and therefore they're more filtered. So, and UV being a cancer causing agent, then there is less need for a shield, for national shield up in the Northern Hemisphere. And that's why Northern Europeans tend to be more pale in their skin because they have less need of melanin. Another example of evolution, right? Especially if the origin was out of Africa, as it is commonly believed. So essentially, the Europeans have lost pigment. But anyway, the freckles are a concentration of pigment, of melanin in certain parts of the skin, in certain skin cells. So just compare, for example, and so that is a physiological thing because the melanin actually expands or contracts when it's exposed. Why do we get a suntan when we go out at the beach or exposed to the sun? Because the melanin with light expands, it is photoreactive. And it expands to try to shield, it's like a little tiny umbrella, essentially, that opens up with light, okay? It's photoreactive. That's a physiological process, right? Okay, so if there's a difference in the freckle pattern between Claire and Laura, that would point to a slightly different physiological process occurring in their melanin. And when we compare the freckle pattern on the noses, they have a slightly different freckle pattern, not only on the noses, but also on their cheeks and their chin and so forth. In general, it seems to me that who has more freckles? Claire and Laura. Laura. Just at face value, it seems like Laura has a little more freckles overall than Claire. So that's pointing to a physiological variance in the background to those freckles because that's melanin, all right? Okay, moving forward, what do we have? At the molecular level, well, I'm sticking with the melanin for the molecular level because physiology is at the level of the whole organism, right? But when we're actually talking about melanin and how melanin is distributed on the skin, we can also assume that there was a molecular underpinning to that melanin distribution on the epidermis of these two ladies, okay? So the same, just at a different level. Physiology would be more at the tissue and organ level and the whole organism, but the background to that deeper layer would be a molecular variation between the two. And finally, the behavioral, okay, they obviously chose different dresses for that day, for that photo, and even though they seem to be holding the same certificate, so the certificate looks like monozygotic also, whatever they're showing there. It looks identical, the two papers that they're holding, okay, but the dresses are certainly different, so they chose different dresses. They behaved differently when they dressed this morning, right? They had different choices of dresses. So that's a behavioral difference right there. Simple as that. They elected to choose different dresses, and that's a behavioral thing, right? Okay. We see that in nature also, right? We see it in the human population. These are all human children, and they're obviously all morphologically different. We could say physiologically, etc., and behaviorally also. Some are saying hi, some are not saying hi. Some are looking at the camera, some are not looking at the camera, etc., etc. Okay, in nature, here is a pride of, well, the tulips is very obvious that they have a variance within the same species of tulip, right? This is in lions, in wild cats, it's called a pride, a group of them, a group. The pride, which tend to be small, but real. So here we have how many lions are in this pride. 4, right? Don't forget this little fellow in the background here. There are 4. This is the mom lioness and 3 cubs. So variance, for example, there was, even though they do look very similar, but for example, there's a spot that I was looking at on the ear. This fellow seems to have a larger black spot than this one on the ear. Okay, just one example. And that could have a consequence. It could be totally innocuous for the species, or it could have a consequence in their behavior. For example, when they move their ears forward, typically in attention or in alert or in danger. You can see the lioness seems to be looking at the camera, right? Probably has smelled the photographer. And you notice that she has, her left ear is forward and her right ear is backward, okay? They can move their ears independently. And she's probably trying to listen to what the camera person is doing, but also keeping an ear because her eyes are focused on seem to be focused on the camera person, right? And so she's got her eyes, her snout, and one ear focused on the camera person, but she's got this other ear focusing laterally just in case there's something coming from the side. Seems the way, interpreting behavior from the morphology. So it could be that that black spot has something to say to another animal that would be coming toward this one, okay? So there could be a selective advantage to having a larger black spot, or it could be a selective advantage to have a smaller black spot. I don't know. Black spot may have a selective consequences. At any rate, we see that there's a variation there. Just pointing out how variants occur in nature. This is what we call the range, all right? A range. Now, we need to talk a little bit about statistics here, again, just to be on all on the same page. This is known as a normal distribution, okay? In a normal distribution, most individuals within the population cluster around the average, the mean. The average is also the arithmetic mean. So for example, when we look at height, the average height for human, we have to distinguish between men and women because we have this slight polymorphism, moderate polymorphism with regards to height, whereas average men tend to be slightly higher than average women. And we also need to talk about the different age by age group, because obviously a child of boy or girl is going to be shorter than an adult boy or girl, right? So the age also makes a difference until we reach our maximum height. But let's say that the average woman is what? I'm going to say about 5'6" or 5'7", right? And the average man is maybe 5'8" or 5'9", it's a couple of inches difference. Most of women at that age and most of men at that age are going to cluster around the mean, all right? And then there will be a few individuals who are taller and a few individuals who are shorter. And the further away from the mean, the fewer individuals that we have. Some who are extremely tall, like, I don't know, the Maasai in Africa, again, the huge, tall fellows. They're at least 7 feet tall or taller, and they make for good basketball players, right? Or on the short side, also happens to be in Africa, the pygmies, which can be less than 4 feet tall, almost half of a Maasai, right? They're not good basketball players in general, and so forth. So those are the tails of the normal distribution, which is also known as a Gaussian distribution, because this guy Gauss is the one who came up with this probabilistic. And it's also known as a bell-shaped curve, right? So normal distribution, Gaussian curve, or bell-shaped curve, the same thing. Many populations in nature and many variants are distributed in a normal distribution. Some are not, but many are in nature, okay? Whether we're talking about height or any kind of characteristic. Now, another layer of complexity is what we call polymorphism. Polymorphism is different shape. So it's not just the height. For example, we have here dogs. How many species do we have? Well, first of all, how many individuals? 2, 4, 6, 8, 10, 12. 2, 4, 6, 8, 10, 12. Okay, so we have a dozen dogs, right? We have a dozen dogs. How many species of dogs do we have? One species. Canis lupus, by the way, is a subspecies of the wolf. All right, a subspecies of the wolf. All right, so we have one species, one substance, one dogness. All right, and 12 individual dogs. What makes them different? Their polymorphism. The polymorphism is not just the size, the width, the height, the shape. It's also the shape of the face. What do we call these polymorphisms in dogs? We call them breeds or races, right? But breeds, basically, all right? Breeds. But because they all belong to the one species, in principle, all of these dogs could mate and produce fertile dogs for offspring, all right? Now, assume, I don't know, really, they seem to be pure races. If these are all pure races or pure breeds, then the interbreeding would be a mutt, right? Or an interbreed. But really, in principle, they could all interbreed if they can figure out the physics of it, all right? The mechanics of it. But let's assume that half of them are male and the other half are female, they could interbreed because they belong to the same species. And that's the definition. Remember the definition of a species, right? Of individual belonging to a species, that they can interbreed and produce fertile offspring. Meyer's biological definition of species, all right? But they're obviously very, very different in their shapes, in their morphology. And they have polymorphism. So it's not just the size, the height, but the amount of hair, the color of the hair, the shape of the face, etc. You can take it down into the internal metabolism also of the different breeds. Some may be able to digest more stuff than others. Some may be very finicky about their diet, etc., etc. All right. So you can see that the 4 levels, the morphological, physiological, molecular, and behavioral applies. This is kind of a neat example because we have so many different breeds of dogs in the world today, but originally all came from some domestication of the wolf. I think it's debated whether it happened in ancient Egypt or in Mesopotamia somewhere, but somewhere in an ancient civilization, they managed and they dared to domesticate a wolf, all right? All right, so those are polymorphisms. The background, and these are also phenotypes. So polymorphisms are phenotypes because they are an expression of the genotype, of the genes that are in there, okay? So for example, they all have eyeball genes, right? But how that gene expresses, there's gonna be variant. Let's say one that is more obvious. They all have ear genes. They all have ear flap genes, but how those genes express is very different from very attentive ears to very droopy ears, okay? But they are ear genes. And so there have to be polymorphisms in the genes to cause the standing ears or the droopy ears. But they're ear genes, right? Okay, so we find polymorphism even all the way down. That would be an example of what kind of polymorphism or variant. Well, we're looking at the genotype of the ears.
Molecular.
Molecular, right? Molecular. All right. Not all distributions are normal distributions. Just for the sake of completion, we have skewed distributions. Some are known as positive or negative skewed. And this is counterintuitive because people see this skewing to the left. And so they think it's a negative skew, but no, it's a positive skew. A positive skew is left of the normal because that's where the larger values are. Okay, and a negative skew is to the right because that's where the smaller values are. Another way of looking at this is where the tail goes, not the bump, but the tail. So if the tail goes to the right, it's positive, and the tail goes to the left, then it's a negative skew. It's just a standard convention. All right, so for example, here is an example of butterflies, heliconian butterflies. They don't all have the exact same pattern. This would be like a skewed distribution. Okay, some look more like what is known as the tiger pattern. Others don't have the tiger pattern, or it's not as evident. Also, there are other distributions that are known as bimodal. Bimodal would be that there are two humps or more, trimodal, quadrimodal, etc., polymodal, all right? Depending on what characteristic we're talking about. If we're talking about the height of humans, again, at the same age, because we have to adjust for age, right? It's not fair to compare a child height to an adult height because they're gonna be different by age. So at the same age, more or less, we can compare the average height of men and women, and it's going to be slightly different. Within the men and within the women, it's going to be a normal distribution. But if we look at them together and we say the average height of people at 1 year of age, at 5 years of age, at 10 years, at 20 years of age, or 30 years of age, we actually end up with a bimodal distribution, okay? If we look at people as a whole, not distinguishing between male and female. So the composite would be bimodal. It's just another example. So drilling a little bit into probability again to be on the same page. Some of this is very basic and elemental. I apologize for that, but I want everybody to be on the same page, okay? Because of the various backgrounds of the cohort. So the number, any number can be expressed in three ways. Any digit, it can be expressed either as an integer with a decimal, or it can be expressed as a fraction or a percentage. And I can tell you, and I'm just going to leave it at that, that is essential that we are all very conversant in going back and forth mentally, almost automatically, between these three numbers that mean the same quantity. So 1/2, 0.5, or 50%, all right? It's very easy with this one. It's probably the easiest one to do. But it's not as easy already when we talk about, instead of 50%, if we talk about 5%, going this way, 5% is not 0.5, it's 0.05, okay? It's 0.05, and it's not 1/2, it's what? 5% is what fraction? 1/20th, like a nickel to a dollar, right? And so it's adding zeros in the denominator, basically, which makes the fraction smaller. All right, so please be conversant with this and you can practice in your mind. Some are very easy, but some are harder. But it's important to be able to do this, at least in the order of magnitude. Where is it that we are in the order of magnitude? Are we in the tens, in the hundreds, in the thousands, etc. Okay, so probabilistic system is based on percentage. But here's a challenge that medicine, for example, the practice of medicine is a probabilistic science. It's based on statistics. In other words, a doctor will never tell us, oh yes, you're going to heal from that illness, or no, you're not going to heal from that illness. They're going to say, well, there's a 95% chance that you will heal if you take this medication. Or if you stop doing that, harmful behavior, there is also a 95% chance that you will heal from the injury, okay? It's, in other words, it's probabilistic. And that probability is based on previous cases. When previous patients stopped hitting their heads with a hammer, you know, the headache went away. And so there's a good chance, but there's a probability. It's based on previous trials. What if no one had ever hit their head before with a hammer? Then we don't know. You will be the first one to heal, and then you'll start the statistic, okay? And the more statistics we have, it's called a statistic, the number, the more robust that probability becomes, okay? And that's precisely the clinical trials that are being done right now as we speak with the vaccines of the COVID, right? In the meantime, they produce already millions of vaccines or vials, just in case it does work, third level of clinical trial, then to get it out in the market. If it doesn't work, that's going to go into the garbage. But just in case, it's out there, it's ready to be distributed in case it works, all right? The third stage of clinical trials. So not only medicine is a statistical, it's a probabilistic science, but the problem is that we are not probabilities. I'm not a probability. I'm an actual determined real life human being. And so the challenge is forever going back and forth between probability and determinism, or the probabilistic system and a deterministic system that we all are, all right? Because let's say with this cancer treatment, with this chemotherapy, there is a 95% chance that I will heal of my cancer with this chemotherapy. But until or unless I go through the therapy, I will never know if I'm on the 95% or on the 5% until I go through it, you see? So it's post facto. That's why Meyer also makes that claim that evolution, whenever we look at it, it has already occurred. And so we have to infer it. We have to infer it from the evidence, you see? And so this is the challenge, that probability, it can be on either side, right? Why do people buy the ticket of the lottery if there's practically nil chance that anyone will get that ticket, get the winning ticket? But somebody has to win it, right? And like they say, you're certainly not going to win it unless you buy the ticket. And that's why people buy it. But the vast majority, literally millions of people, will not get the winning ticket, okay? But they're paying into it. All right, so this is the average. And the mean, the straight up arithmetic mean, there are many different types of means, or several different types, but the normal mean that we talk about, the average, is the arithmetic average, is the sum of the individuals divided into the total number of individuals. And that's how we get the average. A range is all of the individuals in a population. That's the range. So we have to see sometimes there's a narrow range and sometimes there's a large range. For example, between these four individual cats, lions, there is a narrow range of morphology, all right? But in the dogs, what are the dogs? There is a broad range, even any four of them, there's a broad range in that population, of any 4 in here, okay? Certainly a much broader range of dogs, of individual dogs than individual lions. You see, okay? Okay, so that is the range. All of the individuals within that one population. And a population meaning of all the same species, all right? In a particular geographical region. And the variant then is each individual within that population. That's the variance. So this is just technical terms. Let's move forward. Let's look a little bit at genetics because we need to talk about genotype, phenotype, and then genome as a whole, all right? So the genotype is a genetic makeup. What do we mean by the genetic makeup? Literally the base pair sequence on the DNA molecule, all right? The base pair sequences, the nucleotides, the ATCGs on the ladder, that is the genotype, which in clusters, they form genes, and the genes are the ones that may or may not express in proteins. Whereas the phenotype is the observable characteristics. They derive from the genotype. So these 4 levels of phenotypes, you know, behavior, by the way, is also a phenotype. The way that animals and plants behave is a phenotype. How do plants behave, for example? When I say that the plant has a positive phototropism, let me just open another. Where is open? No, new. Yeah, make it big. Positive phototropism. Can you see, is that big enough? Slightly. All right. My suggestion, when you get a challenge like this, that phrase that is kind of loaded with scientific words, you can start from the bottom, from the back, because that's where you're going to find the noun. It's a tropism. Tropism is a movement. It's a movement. Photo is a reference to light somehow, photons, right? And positive, so it's a positive movement toward light. That's what plants have. The shoot of the plant, what sticks out of the ground, right? Trunk, branches, twigs, leaves, they're constantly reaching out toward the light. That's a positive phototropism. That is a plant behavior. They don't have a brain, but they can still behave. Okay? How about the root system? What kind of phototropism does the root system have? Positive or negative? Negative, exactly. Have you ever seen an orchid near a window? The twigs of the orchid would go toward the light, but the roots would go away from the light, away from the window. So that's a negative phototropism. How about if I change the stimulus from light to gravity? Whoops. And I call it a geotropism, reference to Earth, Gaia, right? So what part of the plant has a positive geotropism? The root system. And so for the trunk, what kind of a geotropism does the trunk have? Negative. Right? Okay, so that's plant behavior. Just because they don't have a brain doesn't mean they can't behave. The genome is the whole sequence, the whole DNA sequence, all right, of the human or any species. That was a big project from 1990 to 2000 to decipher the human genome, right? It's exciting times. I was beginning to do my PhD at Purdue, the second PhD in genetics precisely, just when the human genome was deciphered, the whole 3 billion base pairs. It was first 99.99% accurate, which means that there were several million bases still missing. And then Francis Collins, thanks be to God, got the project to extend a little bit. They needed about 1 or 2 more years to finish essentially the entire DNA sequence of the human. But at the end of the day, you know how many, what was the sample population for the human genome? It was about 100 individuals at the end of the day of the 7 billion that we are. And what if those 100 individuals were very variant, were extremes, you know, were super tall or super short? That would be not representative, right? That would not be the average, the mean. So anyway, they chose 100 individuals. I don't know if all the names are known actually, but those are the ones that represent the whole human population. Very, very tiny percentage of 7 billion, all right? Close to 8 billion now, but back then it was probably around 6 billion. Anyway, that's the human genome. It's out there for free to be able to analyze in detail down to the base pair sequence. So the genotype starts with, when I'm talking about the base pair sequence, these are the bases or nucleotides, adenine, thymine, guanine, cytosine, etc. They're complementary because the opposite, A, also always pairs with T, and C always pairs with G. An easy way to remember that is that the straight letters pair with each other. A and T in capital are straight letters, and the curved letters pair with each other, C and G, all right? That's an easy way. And that's why we call that the DNA strand is complementary. And that's why it can be repaired, because if the sequence is mismatched on one side, there's fidelity on the opposite side, all right? And so they're complementary strands. We only need one strand to know what the complement is. That DNA winds upon itself like the ladder, the twisted ladder, which is known as a double helix. So it's the same thing as a twisted ladder. That twisted ladder then winds around these little balls of proteins that are called histones. There are a total of 4 pairs of histones, and they form the nucleosome, which is the functional unit of DNA. The functional unit of DNA is known as the nucleosome. It's a little ball. At the core of the ball is histone proteins, and then winds around 1.7 times, almost 2 times around the histone ball. And then the DNA goes out to another ball and so forth. So this type of array is known as beads on a string like a pearl collar, for example, right? Beads on a string. And then from there, so we have, that's all the second level of coiling. The first level of coiling is the DNA helix itself, the twisted ladder. That's the first level of coiling. And then it coils around the histones, second level of coiling. Then those beads on a string coil upon themselves also. It's like taking a rubber band and twisting it. You know how it starts to wind in like that, a cluster, all right? Forms a wad, a ball, okay? So it winds upon itself, and then that also winds upon itself again and again and again, and that's called supercoiling. And that's how DNA compacts into the cell, all right? And the level of compaction, if we were to stretch out a DNA molecule from our genome, from the chromatin, in other words, all 46 chromosomes in a single molecule, we were to stretch it out down to the DNA, to the double helix only, and get rid of all the supercoiling, all right? Just the double helix stretched out like this, how long would it be? Anybody? Well, 2 meters or 6 feet, all right? So 3 feet is more or less from the hand to the opposite shoulder. That's about 3 feet or 1 yard. 1 yard, which is about 1 meter more or less, okay? So 1 meter is about 3 feet or 1 yard. So 2 meters. 2 meters or 6 yards, 6 feet would be the length of DNA chromatin from every one of our cells in the body. That's a lot of DNA. All that is compacted into the nucleus of a cell. The cell is already microscopic and the nucleus is super microscopic, and inside the nucleus is that 6 feet molecule, all right? That is called compaction, all right? Compaction. And the ratio of compaction is how much? Anybody know? It's about 10,000 times. 10,000 times infolding. That's the level of compaction. So for example, if I fold this paper once, that's one compaction there. That's folded in half, folded again, then that's 4. And then now I have 8 pieces of paper. I keep folding, 16, but the folds, let's see. Okay, so that's 1 fold, 2 folds, 3 fold, 4 fold, 5 fold, 6 fold. I challenge anyone to fold the paper beyond 6 times. That's 6 times. Well, the DNA is compacted into each one of our cells 10,000 times, folded upon itself. Okay, that is compaction, and that what we call that is simply elegant, and that's why most of the DNA is shut down most of the time. It's important that most of the DNA is shut down most of the time in our cells, in most of our cells. Why? Because we have the entire genome in every single cell. So we have all of the genes, the 20,000 genes, maybe as little as 10,000 genes that code for the whole human body in every single cell. In other words, in our liver, we have skin genes, we have brain genes, we have eye genes, we have toe genes in our liver cells. And in our brain cells, we have liver genes and we have kidney genes, all of the genes in every single cell. And so it's important, it's essential that the DNA is shut down in our cells. Otherwise, if all those genes express, it will be absolute chaos. You know, we've had livers growing out of our head and we've had kidneys growing out of our toes. It'll be insane. Obviously, it'll be chaotic, not viable, right? So think about this. Most of the genes in most of our cells are shut down most of the time. And what happens is that the replication machinery or the transcription machinery gets in there, which is a series of proteins, enzymes, yes, and they wedge themselves in to open a little segment of the genes that are going to be read for that particular organ tissue that needs to work, all right? And so it is, and all this is regulated by regulatory enzymes and machinery, which is mostly proteins that are doing that work, okay? And that's why it does function, it does work, because only little segments of the genome are expressed at any one time in any one of our tissues according to the proper function. Okay, so for example, the protein expression is the genotype, is a phenotype, all right? Meaning that, for example, the different types of ear, this happens to be a Mendelian characteristic, either we have it or we don't have it. In other words, our earlobes is either attached or detached, all right? From here I can see, for example, Jordan has a detached earlobe. Detached also, most people have detached. What do you have? Rogelio, detached also? I think I have attached. Anyway, it's either attached or detached, okay? Nothing to it. I don't know, I don't want one or the other. But that's a phenotype, that's an expression from a gene, you know, the earlobe gene, wherever that is on the genome. But the most basic level of phenotype, what is the most basic level of phenotype? If a phenotype is the expression, what was that? The phenotype is the expression, the observable characteristic of the genotype. The most basic phenotype is the protein sequence that comes out of the mRNA, the polypeptide that comes out of the mRNA. You see? That's the most basic phenotype. It's the polypeptide sequence that comes out of an mRNA sequence, right? And then from any level, all the way out to the functional organism, you can take it as far as you want, as far out as you want. Okay, so we're clear about what is the genotype, what is the phenotype, and what is the genome. You know, many genotypes don't all express, and they certainly not all express at the same time, right? So the phenotype may vary along the way. And there are genotypes that never express, and that's a good thing. For example, we have proto-oncogenes, we have oncogenes, we have cancer genes in us, right? There are sequences that code for chaos, for unrestricted growth of the cell. Basically, those genes would inhibit cell regulation to stop undergoing mitosis. And it's a good thing that those oncogenes don't express, right? Otherwise, we end up with cancer when they do express. And there are certain things that trigger those genes to express, like, for example, UV, or sometimes high heat, or other carcinogens, like, for example, nicotine, or maybe caffeine, and so forth, right? So some genotypes are best left alone because they express negative phenotypes. Okay, now so this is an example of the genome itself. The genome is the entire makeup of all the genes in the body, right? And again, a little bit of nomenclature. You know that we have 46 chromosomes or 23 pairs, right? But the 23 pair is only really true in the female, because in the male, the 23rd pair, okay, so 22 pairs are called autosomes because they do not code for our sexual characteristics, right? Our sexual characteristics are only contained in one pair of chromosomes, and they're known as the sex chromosomes. So the other chromosomes that are not sex chromosomes, the other 22 pairs, are known as autosomes, auto, okay? Autosomes. So the autosomes don't have the sexual characteristics, and the sex chromosomes do. And these are the sex chromosomes here. In this particular karyotype, this is called a karyotype, where it shows all of the chromosomes by size and by pair. The 23rd pair, in this particular characteristic, they're telling us is XY. So is this male or female? This is a male, this is a man, okay? It came from a man. So in females is XX. In other words, the same X repeated. So females have no Y chromosome, but they have a second X chromosome. Still, that one of the X chromosomes always has to be silenced. I'm getting too much into next year's lecture, the spring lecture. Anyway, this is called a karyotype, which is the morphology, just the external, the gross morphology. And what it does is they're numbered by size. And so that's the basic genetic makeup of humans. We'll get more into detail, like I say, in the spring course on the beginning of life. Now we're going to talk a little bit about Mendelian traits. I'm going to go for another 20 minutes or so, and then we'll take a break. Mendelian traits. So remember, the counterpart to Darwin was Mendel, right? Gregor Mendel doing the genetics experiments, and he came up with these factors, inheritance factors, which now we know are actually genes. Okay, he did not have the benefit of a microscope or the benefit of molecular biology or anything of the sort. So he just was he going by the genotype or the phenotype? Definitely the phenotype, the express characteristic. But he was reflecting, he was thinking, right? And he was intuiting what is behind, what must be the cause, what is causing these different phenotypes when I am doing my pollination and my hybrid breeds? What is causing the different phenotypes? Because all I'm passing on to the flower, meaning the female reproductive organ, all I'm passing is the sperm, the pollen grains, which is the male reproductive cells, right? And how is pollination done, by the way? Did I mention that? What would he use to pollinate flowers?
It was a tool, like a scraping tool.
Yeah, it's a pencil brush, for painting, right? They dip the pencil brush in the stamen of the flower, pick up the pollen and take it to the other flower and pollinate it, right? And the pistil. And then they cut off the stamen and throw them away and so forth so that they don't contaminate. So it's a detailed work, but it can be done because it's visible. All right, it can be done with the naked eye and patience. And a nice field inside the monastery there in Brno where he could do his experiments. All right, so he was coming up with these characteristics that are called all or none characteristics, all or none, all right? And these are Mendelian traits. To this day, they carry his name because they are all or none. They either show up or not, okay? And at the end of the day, he found 7 characteristics of the pea plant. It is speculated that he must have done some work prior to this and discarded characteristics that were non-Mendelian, meaning that were not all or none, that he would get different height. For example, it's interesting that the pea plant is either tall or short, but there's no polymorphism, or there are only 2 morphisms, the tall and the short. There's no variation, there's no range. The range is made up of 2, either tall or short plants. The tall are all the same size, the short are all the same size. That's rare, that's not common in sizes and shapes, plants or animals, okay? So it's speculated that he must have gone through several different plants and vegetables to find the proper one. But it turns out that for the pea, he found up to 7 characteristics that are all or none. And these are listed here. The most common one is the pea itself is either round or wrinkled, or yellow or green, the pea. But then the pod is either green or yellow. And the pod is either smooth or wrinkled or constricted, right? And then the plants are either tall or short. The leaf junction is a different pattern. Let's see, leaf junction or, oh yes, the flower, where the flower is at the junction of the leaf or the tip of the branch. And then the flower itself is either purple or white. Okay, and so these are the 7 characteristics that he found about the pea. And further, he noticed that the majority of times, these characteristics on the top were the predominant one, were the ones that showed up most of the time. So most, on any pot of peas, most of the peas were round and most of the peas were yellow and so forth. Okay, so those are the dominant ones. And a few times, there were these recessive peas. But it's either one or the other by quality, and then by quantity, more often than not, it was the dominant one. That's what predominated, okay? So he was curious, why is one dominating over the other? Why is it not 50-50, for example, okay? All right, so he started doing his experiments, and the ratio, the big ratio that he came up with after doing the math and the probability was the famous 3-to-1 ratio, okay? Where he would get dominant 3 other times and recessive 1 other times. So that's a 25%. And then going forward doing the Punnett squares, he figured out that these characteristics were being passed on by the father or the mother plant. And it depended if the mother or the father was dominant or recessive, the offspring characteristics that would show up. But the basic ratio was that 3-to-1 ratio, which allowed him to establish this dominance recessive. Mendelian characteristics are not common in nature. They're not common. They're rather rare themselves because it's like that, on or off, okay? It's like a light switch. The light is either on or off. There's no dimmer. In reality, many characteristics in nature are polymorphic, they're variants, okay? There's gonna be a variant, a variation about the mean. So there will be a range. We have a few Mendelian characteristics in our body, but they tend to be secondary. They tend to be non-essential characteristics. For example, whether we have hair in between the digits or not, okay? We either have hair or not hair. And hopefully, if we have hair, both hands have hair. Okay, so what is dominant and what is recessive? Let's see, Rogelio, you have hair in between your digits? You don't have any hair? No? How about Jordan, do you have hair in between your fingers? Well, on the top of the finger. On the top of the finger, do you have hair? Yeah, okay. Carolina? No? That's interesting. We're 50, I do, but it's supposed to be, so having hair is supposed to be dominant and no hair is recessive. How about curling the tongue? Can you curl? Can you curl your tongue? Hmm? Yeah, most people can curl the tongue. That's dominant. And so on and so forth. The widow's peak also is dominant. The earlobe thing, that's number D there. Let me see, earlobe. Yeah, having an earlobe is dominant, okay? Recessive is no earlobe. And then that finger bending thing also, not bending it is dominant, so forth. So, but we have few characteristics. Also, the blood type is Mendelian because we either have the protein or we don't have the protein. And there are 3 different proteins that are associated with the surface with the blood type, all right? On the surface of the RBCs, of the red blood cell. One of them, well, the most common one is what is known as the RH factor, where either RH positive or RH negative, right? Because that's a particular protein on the surface of the red blood cell. We either have it or don't have that protein. To my knowledge, it doesn't have any particular function. We just either have it or don't have it. Okay, so there's no selective advantage that I know of the RH factor, positive or negative. And RH, where does it, do you know your RH factor by any chance? Do you know if you're positive or negative? You're positive? Yeah, I'm negative. You don't know? Okay. Typically, when you have your blood type, you get the blood type A, B, or O, and then positive or negative, right, for donation. Simply, RH comes from the rhesus monkey, okay? The first 2 letters of rhesus is the RH factor. Because it was discovered in this monkey, it's a protein that the rhesus has in its RBCs. And the rhesus monkey is very similar in metabolism to the human, so rhesus monkeys are used in the lab for experimentation before you go to human clinical trials. And so that's the RH factor was discovered there. In reality and in nature, most characteristics are not, most variants are not Mendelian. They're not Mendelian variants that all or none. Most variants, most morphisms are polymorphic. There's a range, okay? So for example, we look at the faces of cats. These are all the same species, but very different faces, right? Very different morphisms of the face. For humans, pigmentation is also a polymorphic trait. You can see from very pale, maybe she's Norwegian, I don't know. But you can see to very dark is the melanin distribution on the skin is polymorphic. For heights, these are giant ferns, tree ferns, probably from Australia or New Zealand. Their heights of these are mature tree ferns, which are rather odd because they only have an elaborate vascular system to get this high, this tall. There's also a range of a polymorphic range. male guppies in the fancy tail and so forth. These mushrooms, these all happen to be the same species of mushrooms. Actually, these are all boletus, which generally are edible, by the way. These are all boletus mushrooms. I know because they have the underneath of the cap is spores instead of gills. Okay, these are all boletus, same species, but look at all the variance. Tremendous variation on the same species of mushroom. Okay, so most characteristics in nature then are polymorphic, and that's why there's so much variation within the species, All right, so this is individual variance within the species. And selection is going to act on variance. Could be a selective advantage to being darker if one is living in Africa, because one is going to be less prone to skin cancer. And some skin cancer can kill you, especially if there's no medicine available, like a few thousand years ago, not to mention 100,000 of years ago. All right, now this leads us to 5 theories of evolution from Darwin, 5 theories that come together within the cluster of evolutionary process. First of all, this is from observations in nature, observations. The non-constancy of species. In other words, species are evolving all the time. It's a dynamic system that is going on within the species, to the point that Darwin actually has difficulty with the concept of species as a whole. Of course, it's an anachronism. You know, we didn't have the biological definition of species from Meyer back in Darwin's time, but it's not that Meyer invented the species. Meyer is proposing a definition of species, a biological definition of species. You know, species were around in Darwin's time. In other words, it just wasn't systematized the way that Meyer gives us that the possibility. But the Linnaean system classification already existed a couple of centuries before Darwin. All right, so the species were already being named by genus and species, right? The binomial classification. But he says it's an unconstancy, so he has difficulty accepting the concept of species as a whole. He'd rather look at variance, Okay, so he would see kind of a continuity between a cat, a domestic cat, you know, a pussycat, and a tiger, and a lion, and a puma, and a panther, all right, or a jaguar. He would have difficulty seeing them as different species. He would say, basically, they're variants. Okay. All organisms descending from a common ancestor. This speaks of branching evolution, or what we call the common ancestor, the nodes on the branching, the nearest common ancestor. Also, the gradualness of evolution, meaning that if a train is pulling wagons, if a motor, the locomotive is pulling a train of wagons, there are no missing wagons in between, all right? There are no missing wagons. There's a constant link between species, between the living species and the extinct species. Our problem is that we just haven't found that fossil, or that fossil doesn't actually exist, what is known as the missing link, because we just, there were maybe just a few individuals of that particular species, of that link. And what happens with those individuals, they got caught up in a volcanic explosion and were burned to ashes, and so there's no actual fossil made of them, right? And he speaks of the paucity or the scarcity of the fossil record. We really haven't found all the fossils that are to be found. Many of them are still in layers underneath the crust of the earth. Could be buried a few inches down, or a few feet, or a few hundred feet below the earth, and we haven't discovered it, all right? So that's also why whenever a fossil is discovered, it's such a big to-do, because it fills in a little bit more, not a little bit of the puzzle, is a piece of the puzzle is found. But certainly fossils are really in general very, very scarce as far as all of the organisms that existed in ancient times. Multiplication of species, this speaks of biodiversity, this branching effect, right? That we see species from one species, several others eventually emerge. Diversification, speciation, typically 2 or more different species come out from one, okay? And all species eventually are doomed to become extinct. No species is going to live forever. We haven't experienced any species to live forever so far, okay? Some may be very successful currently, but invariably, because the climate is also in a dynamic interaction over long periods of time, we have had this pulsation of winterization and tropicalization of the Earth, then at some point the climate is bound to change significantly enough to eliminate any other number of species. This is actually going to open up niches for new species to emerge. Like it happened when the dinosaurs disappeared, we mammals were able to emerge because we just didn't stand a chance with those dinosaurs around. They were just too voracious for any mammal to increase in size significantly. Plus, the Earth was also too tropical for developing a fur, which it was retaining too much heat to be successful as mammals in a tropical Earth. All right, and the whole process of natural selection really, which is the nutshell of it, is the very core, the phrase of natural selection. This is really the pressure, the selective pressure that acts on variation. So essentially the process of evolution is a selecting out. It's a selecting out the less fit, all right? All right, so I'm just going to give you very briefly examples of each one of those points of 4 of the 5, because then natural selection is going to have its own lecture, which will be next time. So I'm just going to give you briefly one example of each one of these. The non-constancy of species is this evolving, the continuous evolution that we see. For example, there's the elephant today. We go back to the mastodon, also known as the mammoth, of which there is evidence, not just fossil, but actually frozen, dead frozen mammoths that have been found in Siberia, for example, frozen into the tundra, into the permafrost with actual tissue, right? And yes, they're trying to clone a mammoth genotype into an elephant egg to see if they can get a live mammoth. So far it's been unsuccessful, but they've tried because they found actual tissue of the mammoth. We go back further, we found fossils of creatures that have been reconstructed. What they do with the fossil, which is typically a skeleton or a skull, the skull, which may be fragmented, can be completed by computer tomography and figure out what was missing on the skull and fill it out, right? So we get the full skull. And then anatomists are able to put, with clay, mimic the muscle structure of those bones, right? Especially when there's an articulation, they can figure out what kinds of muscles would be there. So they do that with clay model on top of the skull, which itself is a model from the original. And then once they have the muscle system, then they put an adipose layer, again with different colored clay on top of that. And finally, on top of the adipose layer, they will put a skin layer, and then they may put a hair layer on top of that skin to see how that fossil may have looked at the external morphology. So it's very detailed work. These are experts that spend their whole lifetime doing this. And so this fossil, for example, may have looked something like this. And the ancestor to this guy was something like this, of which there is a fossil here, a skull. And we can see that the canines were very, very small. All right, the canine teeth, which gradually became larger and larger. This is known as runaway evolution, similar to the tail of the peacock, the male peacock. The peahen is selecting on that tail and she's fixated on the bigger tail, all right? And it can never be too big for her, that tail. And so she will always choose the male with the largest tail. Somehow, this is called runaway selection. That's an example of sexual selection, which is different from natural selection, because natural selection implies random mating. Not sexual mating is not random, right? It's selecting on some particular characteristic. So that is thought what had happened with the canine teeth of the ancestors of the elephant and the mastodon, the mammoth, that eventually became huge tusks, right? That have now a very different function from the canines. And this little guy, the Moeritherium, had very different function for these canines that the elephant today has, right? Okay, but there's a continuity there. The descent of organisms from common ancestors, I've mentioned this many times now. The nodes are the meeting place of common ancestor, and we can go back precisely looking at percentage. How can we construct a phylogenetic tree like this that has all of the three main domains, the bacteria, the archaea, and the eukarya? This includes all the animals, plants, living organisms in the world, these three-branch tree. What can we compare between all of these organisms that are living today to construct this branching tree? How we can, in other words, what I'm asking is the question, how do we know that these diplomonads are more ancient than the ciliates when we're going to the animal branch?
The strata, where they're found within the strata?
The percentage homology of proteins. We don't even need to go down to the DNA level because we know that the genome is expressed in the protein sequence. So we can do protein genetic analysis, protein sequencing, right? Amino acid sequencing, and see percentage homology between different species. For example, we start with flies that look similar. We can take the housefly and the fruit fly and see where they fall on the cladogram. And then we can go back and take, for example, a caterpillar, grasshopper, and compare the grasshopper with the fly and so forth. And we can construct these phylogenetic trees just by doing percentage homology of proteins of different species across the board. And you can spend a lifetime or several lifetimes doing that. Okay. Okay, so that points to a descent from common ancestors, all pointing toward a common root. Finally, the third one is the gradual nest, no saltations. Again, we saw the example of the Eohippus evolving into the common horse today, Equus. Took about 50 million years to do that. And these herbivores were seemingly so abundant, have been so abundant in the prairies of Europe and North America and other parts of the world that sufficient fossils have been found to put together a pretty accurate gradation from one fossil, from one species to the other, so that we have a fairly good continuity of that middle finger of the Eohippus eventually developing into the hoof of the horse of today, literally walking on his, the nail of his middle finger. And the other digits gradually become degenerate, become vestigial, all right, receding in. And the middle digit becoming reinforced because it's holding the weight of, or at least 1/4, each one of 4 digits, 1/4 of the weight of the whole horse, okay? So for very few species, we have a more complete fossil record of transition, but for most species, we have pockets, we have blanks, the missing links, But that doesn't mean that there was a skip in the evolutionary process, okay? It just points to the paucity, the scarcity of the fossil record. Okay. The other point, the multiplication of species, the branching effect. This seems kind of redundant here. We see this is just on the dinosaurs, for example, going through 3 eras, the Jurassic, the Triassic first, then Jurassic, and finally the Cretaceous. For some reason, the Jurassic got the bad fame of the dinosaurs, Jurassic Park, for example, but in the Cretaceous, we actually had more species. It's just that the expansion occurred during the Jurassic, you see? The Triassic was relatively fewer species, but the expansion occurred during the Jurassic, but the full development was more the Cretaceous, and eventually the decline, a fairly rapid decline, which is pointing to some kind of catastrophic event, all right? Because this is in a time sequence here. There's a time sequence horizontally. It doesn't actually give us a scale, but you see there is a relative distance between the 3 points of origin and extinction. And you can see that here is pretty catastrophic. Essentially, all of the dinosaurs were wiped out. The species that were remaining that did not die earlier, okay, were wiped out here. And so that's why it's believed that this coincides with the meteorite that fell into the earth and buried there near Yucatan Peninsula. It's about 2 or 3 miles deep into the crust of the earth. Imagine the speed of that thing, which had even fragmented through the atmosphere and so forth. It's just insane. That produced a huge cloud of dust and ash that essentially covered the earth within 24 hours with a very thick layer of cloud that essentially killed the vegetation of the earth at the time. Most of the vegetation died off. And so most of the herbivore dinosaurs died off because they were feeding on that vegetation, right? Huge bulk masses of biomass that had to be maintained on her vegetation wiped out. So there go the herbivore dinosaurs. And of course, then the carnivore dinosaurs that are feeding on the herbivores are also going to die for lack of food. They ate the last starving herbivore dinosaurs, the carnivores did, and then they proceeded to die off also. Catastrophic for the dinosaurs, and it was a boom for the mammals, which at this time, again, from the fossil record, the only mammals that managed to live during the age of the dinosaurs, going back about 70 million years ago, were little mouse-like little mammals, all right, that would hide in between the rocks and scurry around. Those were the only ones who managed to survive, because if they got any bigger, they would be eaten up by these guys, and was no chance of really flourishing, okay? So when they're wiped out like that catastrophically, all of a sudden, this mouse came out, hey, there's no dinosaur around. Let's reproduce. And as they reproduce, variants managed to survive because no carnivores were around to eat them, or not enough of them, right? So they outpaced the mice outpaced a few carnivores that survived, that were probably small lizard-like reptiles, lizard-like dinosaurs. And so that was the beginning of the flourishing of the diversification of the mammals to today, fast forwarding 70 million years later, we are now in the age of the mammals, okay? And we are the dominant group of animals on Earth. By size and by location, we have essentially conquered every terrain of the Earth, every biome, whether it's on land or in water. Very successful, thanks to the fact that the dinosaurs step aside and essentially left the niche open for new animals to come in there, and new plants also. All right, so at the end of the day, we have this one, for example, is the mammals, all right? These are mammals. I think it's 28 families of mammals that we have. And this is called expansive radiation from one single common ancestor of mammals, one little mouse-like creature that managed to survive the dinosaurs, okay? One species, the branching effect, gradually, but really. And you can see that most branches of the mammals are still living today. A few have died off, okay? Specifically 1, 2, 3, 4, 5, 6, 7, 8. And this one is Cetacea. This one is dying off. What are the cetaceans? Quails, dolphins, dugongs, and manatees, all of the sea mammals, right? There's an article just yesterday in BBC Worldwide that the cetaceans, the whales, are definitely on the way to extinction. So they had their age way back here. Anyway, these are the mammals of today. We can see definitely an expansive radiation, all right? Expansive adaptive radiation because niches open up that were not available earlier, mostly due to predation. Okay, let me stop and take a little break and we'll come back. Before I do that, any questions or comments? Basically, we talked about variation, the range of population polymorphisms, right? Some genetics, quantitative traits, and Darwinian evolution. Oh, yes, sorry, maybe I forgot or I skipped that. Quantitative traits, where's the slide for quantitative? Oh, yes, okay. Yes, quantitative trait are non-Darwinian, is when there is a range, when you have variance. That's a quantitative trait, okay? And so non-Mendelian traits, non-Mendelian, that it's either on or off, okay, show or no show. The ones that, the traits that show a range of expression of variance, those are known as quantitative traits. It's just the technicality of the language in genetics, right? And most traits in nature are quantitative. And so that's why selection can act on those quantitative traits, on those variants. Okay, now I'll really stop and we'll come back. Any questions, Jorge, Chris?
No.
Comments? No? Okay.
No questions.
Thanks. So let's be back about 10 minutes, just under 12, because I still need to cover a whole set of other slides. Okay. So 10-minute break, please. Let me pause this. Pause. Here we go. Welcome back. For the second part of this lecture, I want to look at 3 of the 4 main forces that actually move evolution, the mechanism of evolution, the how does evolution actually happen. And these are mutation, migration, drift, genetic drift, and then finally, like I mentioned, selection, natural selection, for the next lecture, okay? Let's go forward then. This one is a PDF because I made it into a PDF and I lost the PowerPoint. So I have to work on the PDF eventually. I guess I can rebuild the PowerPoint from this one. All right, so mutations are going to occur at the gene level, the level of genes. In other words, on the DNA sequence itself, right? It's going to be some kind of a mismatch that happens in the DNA sequence. And this is what I mentioned earlier, the human genome, that is 3 billion base pairs. That's 10 to the 9, right? 3 billion of these little letters, give or take a few hundred, a few thousand, which would be variance between all of us or among all of us. These mutations can be, strictly speaking, of 3 different types, either mutations as such or recombination of DNA, especially in sexual reproduction. I'll show you in a minute how recombination plays into the mutation aspect of the genes. And then the phenomenon of transposition or transposons. How many of you have ever heard of transposons? Have you heard of transposons before?
Yes.
No? Okay, we'll get to it. It has to do with the DNA jumping around from section to section. So what occurs, the DNA molecule is subject to mutations, either in sexual reproduction or in asexual reproduction, okay? In sexual reproduction is in the precursors of the gametes. In other words, the organs or the cells, the tissues in the organs that make the gametes, that make the egg and the sperm. That's going to be in sexual reproduction. For asexual reproduction, it's going to be mostly by a couple of techniques called lateral gene transfer, or also in clones. Clones, which, for example, when you look at mitosis, the cells that, the resulting cells are clones of each other. So let's look at sexual reproduction first, and the organs that make those cells, that make the gametes. You know that gametes are sex cells, and they're egg and sperm. So the eggs are produced in the ovaries, the sperm are produced in the testes or testicles, and there is tissue that is the precursor tissue in the ovaries for making eggs, and in the testes for making sperm, all right? That precursor tissue, if we look at a cross-section, for example, of an ovary, are the follicles which have an oocyte or an egg embedded in it that matures in a cycle, and this is the cycle represented here. Here's a follicle maturing and an ovulation process of the mature egg rupturing through the ovary and then going to the fallopian tube for the possibility of fertilization. Okay? In the testes, there are these tubules that are inside the testes proper, the testicles. They're called seminiferous tubules. And these tubules, the inner lining of those little tubes, those are the precursor cells. Those are the cells that make the actual sperm cells, all right? Before they get a tail, then those cells move out to the vas deferens where they develop the tail and so forth. So the process of producing these gametes, whether they're sperm or egg, is called gametogenesis. Gametogenesis, or the production, the generation of gametes. The tissue that makes those gametes is subject to mutation. The mutations can be spontaneous mutations. Mutations are, in nature, they're spontaneous, all right? And most mutations are neutral. Well, first of all, 97% of the genome is non-coding region of the human genome. And so if a mutation falls in that non-coding region, so what? Who cares? It doesn't affect anything. It doesn't affect any genes. But even in the 3%, many times it doesn't alter the gene significantly. And so it's what is known as a neutral mutation, or sometimes it's beneficial, and sometimes it is detrimental, the mutation on the genetic sequence itself. For asexual reproduction, these are just different examples of asexual reproduction. This one is known as binary fission or lateral gene transfer. There's actually a little tube that forms from one bacterium to the other, and a segment of DNA can be transferred through that tube, which is known as a pilus, into the new bacterium, and that is a lateral gene transfer, sharing essentially DNA from one bacterium to the other. In other microorganisms, in other organisms, whether they're small, they're micro or not, the general way of passing some genetic information to either another cell or a next generation is by variants of cloning or mitosis. When the cell splits, and the daughter cells end up with the genetic material that was in the original mother cell. So different types of asexual reproduction, but they're all basically variants of mitosis or cloning at the level of entire organisms. The point is that both in asexual and sexual reproduction, there is some possibility of passing on a mutation to the next generation. Okay, this is a little bit too much detail probably, but there are different types of mutation depending on where it occurs and what is actually mutated. For example, one mutation is changing a cytosine to a uracil. In other words, a cytosine instead of a thymine, it normally is thymine that converts to uracil in the RNA. But here it's a cytosine, so that would make this particular sequence on the codon, the triplet, a stop codon, which will stop this protein literally from forming correctly. It will be fragmented as opposed to the normal beta globulin. These are the proteins that make up hemoglobin, all right? And so this protein will be truncated and deform and not function as well. And this illness, this is an actual illness that's called thalassemia, beta thalassemia, is more prevalent in Europe. And these people typically are weak because they don't oxygenate as well. Their blood doesn't oxygenate, they have a defective hemoglobin, all right? This is an example of a point mutation. It's a simple substitution from one base to another, but it happens to fall in a critical area of the protein. But this is very rare, really. Seldom do mutations become that critical to interrupt a sequence, a protein sequence, right? More common, also we can have point deletions instead of a point substitution, a point deletion, where an actual nucleotide is missing, and then that will change what is known as the reading frame. The triplets are not the original triplets. It changes the reading frame, the reading frame of the DNA, so that now different amino acids are being coded for the new sequence, and it will make for a different protein also, all right? Which typically will be a defective protein. So those are point deletions, but it has to happen in the coding region of the DNA for it to have this kind of an effect. Or it could be that after downstream from the deletion, then the new code becomes a nonsense code. In other words, it doesn't really code for any amino acid. Other examples of mutations, they can be deletions of entire segments or inversions where the segment reverses its sequence, especially if it's palindromic. Palindromic is that it's like a mirror image. The sequence has a mirror image to it, next to it, and they reverse size. Or sometimes the sequence duplicates, the same sequence sticks at the end and repeats, all right? Sometimes they invert. All these are possibilities of mutations that can happen in the DNA sequence at the level of the chromosome pairs. You see, these are the chromosome pairs here. So different types of mutations, whether they can be single point mutation on a single nucleotide or entire regions, small regions, longer regions, There are also mutagens. Mutagens are substances that can cause a mutation on the DNA. And these mutagens are also known as carcinogens because of the potential for causing cancer. The very one well known is the UV. There are three types of UV essentially that hit the Earth, A, B, and C. And the wavelengths are different, and that's why they have different penetration range for the atmosphere. The most damaging one is UVA. So when you buy your sunscreen, you look for the UVA grading. If it's UVC grading, that's useless. You're just wasting money there because UVC is filtered all the way out at the outside of the ozone layer in the atmosphere, okay? B tends to penetrate a little bit, but UVA is the one that penetrates the most to the actual surface of the Earth where we're standing. The ozone is the one, O3 is the one that filters that UV. So UV light can cause, the typical one is these thymine dimers. Two thymines that are next to each other tend to stick, they link, they cross-link. And so it causes a little kink on the DNA and it dissociates from the two adenines on the other side. And so this is going to cause a misread either of the replication machinery for DNA duplication or the transcription machinery for making messenger RNA, right? And so these thymine dimers are typical of UV exposure, can eventually lead to cancer. Other times it is because of certain substances that mimic the nucleotides and can they actually wedge inside the DNA ladder and disrupt the sequence, okay? Those are also carcinogens. They're chemicals, very dangerous. One of them is acridine that you can see here. Actually, that molecule is a 3-ring molecule, fits inside the DNA and causes a disruption on the sequence, on the genetic sequence. Heat can also do it. High heat, because high heat can denature DNA. It breaks up DNA, all right? And it denatures the bonds. First, it breaks the hydrogen bonds in the middle and unzips the DNA. But beyond that, it also can break the glycosidic bond here that fixes the pentose to or the hexose to the rail of the ladder, all right? So bottom line is that high heat can also cause mutations in the DNA. Okay, let's look at recombination for a moment, how that happens. This is during sexual reproduction when the chromosome pairs exchange segments, pieces of DNA. They exchange pieces of DNA. Here's one chromosome pair. They line up at the equator of the cell in meiosis 1, the first division of the first cell division, whether it's in the ovary or the testicles, for the formation of these gametes. And at that point, when the chromosomes are paired up at the equator of the cell, this is called a tetrad because it has 4 legs, 2 from each chromosome. And at the tetrad level, they can exchange, the legs can exchange segments, pieces from one to the other. This event is called recombination. It's also known as crossing over, all right? And then this is where the genes from one allele, one DNA, one chromosome sequence is exchanged with the sequence of another chromosome. And it could be, for example, that the blue chromosome is the dominant allele, the dominant of the pair, and the red is the recessive. So by the time we get to after the recombination event, then after the recombination, this dominant chromosome turns out to have a segment that is recessive. And conversely, this recessive chromosome has a segment that is dominant. So it depends, when the meiosis process completes, right, down to the 4 daughter cells, two daughter cells are going to end up with this. Actually, it will be 4 combinations. One daughter cell is going to end up with this leg of the chromosome. Another daughter cell is going to end up with this leg. Another daughter cell with this one. And finally, another daughter cell with the fourth leg here. Meaning that there are 4 possibilities. 2 are recombinant DNA, recombinant legs, and then 1 is dominant and 1 is recessive. And it all depends which egg or which sperm fertilizes, if it's going to be the recombinant genes or the dominant or recessive gene, that's what's going to express, okay? So recombination is actually a very powerful tool for maintaining variation in the progeny. The big picture on recombination is that it stimulates or it encourages variation in the progeny. And that's really, this event of recombination is common and frequent. And that is why we are not exactly like our parents. In other words, sons are not exactly like their fathers, and daughters are not exactly like their mothers, okay, because of recombination. So daughters get some of the recombination events from their father, and sons get some of the recombination events from their mother to make them have then these quantitative traits, to have variation. All right, here's another. Oh yes, there is also variation inserted in what is known as independent assortment of alleles. The alleles are each individual leg of the chromosome, all right? Those are the alleles. This is maybe a relatively new word. When the chromosomes separate after meiosis II and go to the opposite poles of the cell for making the gametes, there are 2 possibilities. Here we have 2 pairs of chromosomes, 2 large, 1 large pair, 1 small pair, okay? When they segregate, they can either segregate all of the red, both reds together, or 1 red and 1 blue of both the tall and the short. So these are the 2 possible combinations here. This is aside from recombination, this is aside from crossing over. This is the entire chromosome as a whole piece, all right? You can think of these as one being the paternal allele, the paternal chromosome, the other one being the maternal chromosome. For 2 chromosomes or 3 or 4, we can have any combination. We can either get the paternal chromosome or the maternal chromosome, right, of the pair for every one of the 23 pairs. So there is mixture there too. This is known as independent assortment. This is another one of the laws of Mendelian genetics, that he figured out that the assortment is independent of each chromosome, what he was calling this factors, these inheritance factors. The assortment is independent for each one of the 23 pairs. So there's a good possibility there for mixing, for variation to appear. And so depending on whether we get possibility A or possibility B, these are the outcomes of the 4 cells. For possibility A, 2 blue and 2 blue, and 2 red and 2 red. Or for possibility B, 1 red and 1 blue for tall, and 1 red and 1 blue for short. Okay. So there's a mixing event here that is just probabilistic. On average, this will happen half of the time for one set of cells and the other half for the other set. Okay, a different story is this issue of transposons. Transposons are chromosome segments that jump from one area to the other on the chromosome. They jump around, okay? So Again, the chromosomes are in a somewhat dynamic state, even though they are compacted and compressed in there and supercoiled. Even so, these are events that occur that is known to occur, all right, where segments of DNA jump within the molecule itself from one region to the next. It's not really fully understand the mechanism, how this happens, but we have the evidence that it actually does happen. So here's a little video that is illustrating that. Let's take a look for a moment. Short, it's only about 3 minutes. And this could be evidence that we have been integrated DNA from viruses into our genome. We have about maybe 15, 18% of our genome is actually viral sequences or fragments of viral sequences that have jumped around inside our genome. Okay, I had maybe on mute. No, unmute. Try now.
Transposable.
Sorry, I know what I need to do here on the film. Give me a minute. I need to optimize here. Okay, transposable elements or transposons.
Elements, which are found in virtually all cells, are segments of DNA that can move from one location in the genome to a target sequence in another. A simple type of transposable element called an insertion sequence or IS element is only 700 to 1500 base pairs long and consists of two basic parts, a coding region for an enzyme called transposase and two short inverted repeat flanking sequences. The cellular machinery transcribes the transposase coding region and translates it into the transposase enzyme. Transposase catalyzes the transposition process. The transposase randomly selects one of many possible target sequences where it will move the insertion element. The IS element, depending on the particular type, may enter the target sequence by one of two general mechanisms: replicative transposition or non-replicative transposition. In replicative transposition, transposase nicks the transposable element and the nicked ends attack the target sequence, making a staggered cut in the target DNA. DNA polymerase fills in the large gaps, and DNA ligase seals the remaining nicks. A recombination event resolves the two molecules. As a result of replicative transposition, the cell has a copy of the transposable element flanked by duplicated target sequences. Non-replicative transposition is sometimes called cut-and-paste replication. In this process, transposase brings inverted repeats and flanking DNA together. One phosphodiester bond is cleaved on each strand at opposite ends of the IS element, yielding free 3' OH groups. Each 3' OH end attacks the other strand of the double helix, creating a new bond that results in hairpin structures. Host carrier DNA is ejected, and the hairpins on the IS element are renicked and attack target DNA. From the 3' hydroxyl groups, the cell's DNA polymerase molecules fill in the gaps, and DNA ligase seals the remaining nicks. In this way, the element jumps from one DNA molecule to another. To summarize, in non-replicative transposition, the transposable element is cut and pasted from one DNA molecule to another. In replicative transposition, the transposable element is copied to yield two transposable elements, one in the original position and one in the target DNA.
Okay, so transposable elements are very interesting, and we all have them, and it's DNA essentially that jumps around, sequences that jump around inside the entire genome. So over periods of time, these sequences may jump into a region to either disrupt a gene that is there or to change the functionality of that gene. And rarely it may be that it actually enhances the gene to do a better function and so forth, and therefore it would have an evolutionary advantage. Okay? But here's a mechanism pointing at this mechanical aspect of changing the DNA sequence by this process of transposition. Okay. Another possibility of changing the DNA sequence is precisely viral infections, how that works. You know, if you're familiar with the lytic and the isogenic stages, basically in the lytic phase is when the virus infects a cell, all right? So if we start up here, for example, the virus is essentially a crystal of DNA or RNA, like coronavirus is RNA, it's a retrovirus, similar to the flu. That's injected into the cell. And then that RNA or DNA has two possibilities. Either it integrates into the host DNA, or it sequesters the reproduction machinery, the transcription machinery of the whole cell, and starts making more of itself. Essentially, what the virus needs to do is make more capsules, viral capsule, which is it's mostly a protein capsule. And sometimes there's a lipid layer also, just by coincidence, because I prepared this 3 years ago, and this happens to be a coronavirus type or also flu type, the spikes being the reference to the corona or the crown, which typically are retroviruses. And they also have a lipid layer on them. That's, by the way, that's what the hand washing does. That's why it needs to be at least 20 seconds, because it's the amphipathic, is the soapy aspect of the soap that gets into the lipid layer of the virus on the outside and basically strips that lipid layer, and then the virus becomes totally vulnerable and dies or is destroyed. Can't really call it living because they don't metabolize on their own and they don't reproduce on their own. They need a host to do that. So really, they're not really technically living. They're closer to a crystal than they are to an actual living organism, the viruses. At any rate, this phase, when it sequesters, when the virus sequesters the transcription machinery of the host cell to make more of its own, and it will make, you know, hundreds or thousands more inside the cell until the cell then explodes. And from one virus, now we have 1,000 viruses infecting 1,000 new cells. You see, and that's why it ramps up very fast the infective stage and can be very harmful. That's called the Lithic Phase because it lyses the cell, it explodes the cell open. Whereas when the RNA or DNA of the virus integrates into the host DNA, right, then that's called the lysogenic phase because it becomes part of the genome of the host. And it could be there stored for generations. And that segment of the viral DNA will then be passed on to the offspring, to the next generation, and so forth. And that's how we end up with about 15 to 20% of our genome being viral sequences that we humans have picked up over the millions of years that we've been around from nature. Okay, so summary then of the first force, which is a mutation. There has to be a mutation of some sort in order to cause a variant, you see, because again, there's a mechanical explanation for this. The mutation can be either as mutation as such on the DNA sequence, or by the recombination event, or by transposons. And we can see viruses kind of analogous to transposons in that they inject or insert a sequence, a foreign sequence onto the host DNA, all right? Going to move now to the next stage, which is at the organismal level, which is this gene flow or migration or gene flow, all right? So migrations can be seen as immigration or emigration, depending if individuals are coming into a population or exiting the population, right? So immigration is coming into a population, emigration is leaving that population. Individuals of the same species. So think in your mind, right? The definition of population, individuals of the same species. There's a dynamic system going on for any population in the world of humans and non-humans, any species, down to bacterial. The individuals within that population are constantly moving in and out of the population, right? There are variants within that population. For example, here, the zebra, this is a population of zebra, okay? And at first blush, they all look very similar. We can say they're actually identical to each other, but they're not. If we take a closer look, the pattern, the specific pattern in each zebra is actually different. It's almost like a fingerprint, right? You see the pattern on this zebra on the top is different from this one. And this one, again, different from the others around it. So they all have a zebra-like pattern, but individuals have a different pattern from each other. So it depends. Gene flow is that on any given day, maybe it was this zebra that wandered off this population and went into an adjacent population of zebras on the other side. So this zebra brought with himself or herself its own pattern and not the pattern of the other zebra that stayed in population A. You follow? So there's a flow. Let's say there are two populations of zebra, A and B, that are moving next to each other through the prairie, all right? There will be some flow from one population to the other of some individuals. Not all individuals are going to flow in and out, right? But some will. Some populations are more strict or constrained. Others are more liberal in their movement back and forth. It depends on the species and depends on many factors. It also happens with plants. For example, I can ask you, how many populations are visible here? Well, let's start first with a more basic question. How many species of plants can be seen just a plain sight from here? We can say that there are at least how many species of plants here? This is not too, I'm sorry, it's not too focused. Well, it's two species, at least two species. We have these tall species of grass, tufts of grass coming out, kind of individually on the sand. And then we have this cluster of vines, right? These are clusters of vines. You can see some trailings of the vines here, whereas you can see tufts here. So there are these tufts of grass and then these trailings of vines, okay? But actually the vines, even though this little hill here and this little hill there, let's assume that this is the same species of vine. These are two populations because they're divided by a barren area, okay? So we can say this is population A and this is population B of vines, all right? And if some of these seeds of vine B go to vine A and they'll start growing there, they will bring their characteristics from vine B to population A and vice versa. So there's going to be some interaction, but typically less in plants. And for these tufts of grass down here, it's even more hardship. These are actually called pioneers because they're establishing in a new area, a new terrain, all right, that was previously barren, essentially just sand. So there's some migration of individuals from populations, from one population to the other, and that is known as gene flow, okay? So that's also another factor on which selection will act for evolution, for the possibility of evolution. Oops. Okay, so sometimes the ranges are very narrow, sometimes the ranges are very wide. And I have two extremes here. For example, anyone know what these are? These are called sloths. Yeah, they're kind of camouflage. I didn't realize that the resolution is not that good. These are called sloths, okay? And they stick around their range. They hang upside down. They're mammals. They hang upside down from trees, and they're tropical. I've seen them in Panama, actually. They cross the road, and it makes you cry to see them cross the road because they're very awkward. They seem disjointed. They crawl basically on the road. It takes them a couple of hours to go across the road because they're very, very slow. And typically, they only move within a few feet of where they were born, okay, for the whole lifetime. And they go from tree to tree, maybe a few hundred feet, okay, but they stay in the same general area. That's why they call sloth. They move very slowly. And there are two-toed sloths and three-toed sloths, so we can tell that this is a three-toed sloth. It's got its three big claws on it. All right, so this is a very narrow range of an animal, actually, which typically associated with movement. In contrast with a coniferous forest, a forest of pine trees, with the seeds that are airborne. So you see the seeds are winged, right? They have a little wing. And so coniferous forest, a pine tree forest, has a huge range on a mountain range, you know, and yet individuals are cemented or are actually rooted on the place. So the individual trees are not moving around, but the population as a whole has a huge range, right? And therefore, many more variants can occur in a mountain range of pine forest, even if it's a single species of pine that is there, which would make for a single population, all right? So that's with regards to gene flow. Also, on average, generally, the edge of the population is more subject to variation, to flow, than the core of the population. For example, in this patch of daisies here, you see that the core is fairly solid green, yellow flowers, but then at the edge, there's some mixture with some little violets that are coming into this area and some other plants. So there's more mixture at the edge, right? There's more emigration of daisies from the core out and some more immigration of the violets into the daisies and so forth. So typically the edges have more of a dynamic variation on them. It also happens at the microscopic level. This is mold, for example. The edge of the mold is in closer, more interaction with the nutrient of the agar on the Petri dish, and also the edge is two edges that will come in contact with each other and therefore exchange, immigrate and emigrate back and forth between two colonies or populations. These are actually, each colony of mold here can be considered a population, all right? Even if it's all the same species. At the bacterial level also, this is just a macro graph of bacteria. You can see that the edge is a different color. They are somehow interacting with the nutrient medium where they're at. So it stands to reason that there will be more variation at the edge than on the core of the population. But there's dynamic interaction all the way across from bacteria all the way up to whales in any population. Gene flow. So to understand gene flow, also you can consider the organism, whether it's a plant, animal, fungi, or bacteria, as a bag of genes, essentially a bag of genes that is moving around, okay? And so gene flow in and out of populations. Okay, this is succession, basically explaining climaxomania. We'll see this in later in the environmental course. Thank you. Okay, the third one, and we're coming toward the end. The third is genetic drift. So flow and drift are different, okay? Gene flow is the actual individual flowing in and out of populations of the same species, but different areas, typically geographical areas. Genetic drift is a random event. It's a random choice, a random chance, and that's why it's called a stochastic process. Stochastic is a reference to the fact that it is random. Random meaning that it happens per chance that these are the fish that are swimming through this reef at this time, all right? It could be different. So here's a coral reef. There's a photograph of a coral reef. When this photograph was taken, the school of fish that was swimming closest to the camera happened to be these tanks, these blue tanks, all right? But it could have been the chromies, these orange chromies that are behind, that when the photograph was taken, the orange chromies were the ones that was a school of fish that was swirling in front of the camera, all right? So it's a chance event. It was not programmed that way. It wasn't that the tanks were told, okay, tanks, gather up front because you're going to be parading in front of the camera now. See, it's a chance event. And that is called drift, okay, genetic drift. So it narrows the pool of genes that is available, say for mating or for egg laying, to this particular group that is at this spot here and now, and not another group that will come later. Also, another way of seeing it is as sampling error, okay? For example, we have a bottle of mixed marbles. We start with an even amount of marbles of blue and yellow. There's an even amount, and it's a large amount. Then the marbles are going to go through what is known as a bottleneck event, precisely, a bottleneck event or effect. It could be in nature, it could be a corridor, it could be some bad weather that happened, or it could be geographically an actual canyon, for example, that the animals have to go through, or a region where there are predators that are preying on a particular population, lions and zebra, for example, okay? But a bottleneck effect or event is one that reduces the number of individuals of the population, so that only a few marbles came out through that bottleneck of the bottle, okay? Only a few marbles came out, and it happens that more blue marbles came out than yellow marbles because of the bottleneck. It was by chance, it was random, it was not selected, okay? And so what happened was, if these blue and yellow marbles represent individuals in a population, living individuals, they are all going to reproduce. And they're all going to reproduce on average at the same rate. But because this starting population now has more blue than yellow marbles, when we scale that up to the size of the whole bottle, to hundreds of blue and yellow marbles, we're going to have more blue marbles than yellow because of the bottleneck effect. More blue marbles got through the bottleneck effect than yellow by chance. Just like more green beetles got killed by being stepped on than the brown beetles by chance. So when the new population develops, more brown beetles are going to be prevalent, okay? So a stochastic event is a chance event, and it's also known as genetic drift. It also means that, let's say the yellow marbles that did not go through the bottleneck, those genes, those variants were discarded, were lost. The same here. The variants of the beetles that did not survive, the variation that they carried, that was lost, did not go into the new population. Okay, a couple more slides. Genetic drift in nature can be caused either by biotic factors or by abiotic factors. Remember, biotic factors are living factors, okay? Excuse me, abiotic factors are non-living factors or events. So for example, biotic factor is simply the cows munching on the grass, right? And so as far as the neck of the cow can reach, that grass is going to be trimmed at the edge. And so the grass that got eaten by the cow, that variant no longer exists. That variant was eliminated on the grass, okay? The grass variant that got eaten by the cow, that variant is gone. An abiotic factor and non-biotic factor, right? Could be a natural event, for example, lightning of the pine trees that are out there. This pine tree got hit by lightning, so that variant, whatever characteristics that pine tree had, is gone. It's gone. Those characteristics are lost, okay? And that's why generally the more robust the population, the more individuals in the population, and the more variation in the population, in general, the more robust it is, because if any one individual is lost, then there are other variants to make up for that lost variation. Okay? I actually saw this happen about 3 years ago. A pine tree, huge pine that was in the middle there, got hit by lightning. I was in my office with Dr. Plunkett was in the next door office. It was the middle of the morning with a huge downpour in the summertime. One of those thunderstorms, and the lightning actually hit the tree, turned it yellow, orange, bright like a match, lit up in fire, a ball of fire. And the next second, it was gone. In other words, the fire stopped and the tree was just standing there like nothing happened. But little by little, sure enough, it started browning from the bottom. The needles started browning from the bottom branches at the edge, started browning, and little by little over months, every time I went to my office, I cried. The thing was dying. Little, it took about 3 months for the whole tree to turn brown. But eventually it was the biggest one out there. That's what it gets for being big, got hit by the lightning. That's their fate. They grow to 100 and 150 years, and their fate is literally to go out in a lightning strike. A big hurrah ending. So that can also happen. All right, so genetic drift, remember, is a stochastic process, all right? And it will eliminate certain characteristics from the population. Hopefully other individuals have similar variants, but that variation from that specific individual is going to be lost, okay? All right, so these are the three forces on which selection is actually going to act. Mutation is the best, is the first one, not the best one, the first one, because mutation is the one that allows for variation, all right, for the expression of variation. And then that variation is going to be enhanced, if you will, by flow, genetic flow from gene flow of individuals from one population to the other, and also by drift, in other words, random elimination of individuals within the population. So this brings us to, it sets us at the step of going into natural selection itself next time. And basically, that's what I have for today. Any questions or comments or anything? Mm-hmm. All right, good. So we'll be in touch. I'm still trying to figure out one weekend that we can use, one Saturday that we can use to go to the forest. But I'm thinking waiting a little bit until the rainy season is over or starts to peter out, because like this morning, there was another group going out there from the previous cohort. They had to cancel because it was raining. So I don't want to lose a class and then have it rain and lose both the class and the field trip. Okay. So we're just going to wait a little bit. We still have time till December. Okay, folks, thanks again. I'm going to close this recording now, and I forgot to go back to get rid of the optimize. Hopefully it did not affect the recording too much. Jorge and Chris, were you able to see the presentation since the video?
Yep, I was able to see the video.
And heard all right also? Good?
Yes, sir.
Okay.
It was good.
Thank you. So hopefully everything recorded well. Let me then end here.
You have the links within the PowerPoints too. So yeah, we're set.
Yes, yes, thank you. I always try to do that to embed the link just in case, just for redundancy, right? So you have access no matter what.
Yeah.
Okay, thanks again. And
Thank you.
Have a good one. See you next week.
Thank you. You too. Bye-bye now.
Bye.
Recording here. Okay, Jorge, see you.